Hello, Assalamualaikum, and hi everyone. So for today, I will present to you our work with title Impact of Reversible Inhibitor Binding to PAD4 by using the MD simulation and MPBSA calculation. And my name is Helmi Husaini Fithri from Pharmaceutical Chemistry Department of IIUM. So here is the content for the presentation. And for introduction, I will present to you what actually PAD4. PAD4 is a potential therapeutic target for rheumatoid arthritis. As we can see in figure 1, rheumatoid arthritis is affected at the joint. And PAD4 is reported to be overexpressed in RA patients, especially at synovial membrane. And significance of PAD4 here, PAD4 function to convert the peptidyl arginine into the peptidylcysteine. And the peptidylcysteine can become the epitope of SCPA, and this will increase the cytokine and inflammation and lead to the joint damage to the patient. So the significance here, if we capable to inhibit the PAD4 indirectly, we can reduce the overproduction of citrulline. And indirectly also, we can reduce the joint damage for the patient. And structure of PAD4 currently has actin structure in the PDB, and PAD4 has C-terminal and N-terminal. And if N-terminal has subdomain 1 and subdomain 2. And PAD4 need 5 ion for substrate binding. And current PAD4 inhibitor has two classes, which is covalent inhibitor and non-covalent inhibitor. Covalent inhibitors such as F-amidine using the front door to bind at the PAD4. While the non-covalent inhibitor, for example, here is GSK199 by using the back door. GSK compound currently has reported GSK106 as inactive, although GSK199 and GSK106 has similar topology at the head of the compound, but it record a difference in IC50. We can see here that 0.2 micrometer for the IC50 GSK199, while more than 100 micrometer for GSK106. What difference between them is GSK199 has different location of aminopyrimidine group and extra or metacyclic group here. Due to this lack information, impact of ligand towards the PAD4, we need to discover about it. So the objective of this study to investigate the impact of GSK199 and GSK106 binding towards PAD4 by using the MD simulation and MPBSA calculation. Okay, so for the methodology, we will start with molecular docking. So preparation of ligand involve the preparation of PAD4 with each protein, ligand, and parameter. And then we do the control docking and before proceed with the docking. For the MD simulation, we need also prepare the protein, ligand, and simulation. And then we add the solvent. In our case, we add water. And we add the contact ion, which is chloride ion, to stabilize the system. And we do the energy minimization and equilibration phase. In equilibration, we have two phases, which is NVT and NPT. NVT, we want to induce the temperature, while NPT, you want to induce the pressure. And then we run the simulation for 100 milliseconds, and then we analyze the simulation. Okay, for result and discussion, we will discuss first is molecular docking. Compound GSK199, which is active compound, record higher binding affinity, which is negative 9.6, while GSK106 record negative 9.6. Both ligand actually bind at the back door. Although the GSK106 is lower than active compound, it still show a good binding affinity, which is negative 9.6, which we cannot be ignored. And then we run the simulation. After that, we analyze it, and we can see here that MSD of the GSK199, which color in the black, has stabilized at 22 nanoseconds, and it reached the lowest point of compactness at 22 nanoseconds also. Due to that, we expecting the production phase for GSK199 start at 22 nanoseconds, while GSK106 start at 20 due to the long equilibration phase here and long equilibration phase here. For RMSF and big factor, P84 with GSK 199 record a high value of fluctuation at N 3-month subdomain 1 here. We can see in the black color, it has higher value of RMSF compared to GSK 106. And then we can see here as has fluctuation here. It happened due to this structure actually a loop structure. Due to that, it fluctuates compared to the others. And then we can see here that at C terminus has a similar pattern of the fluctuation. For hydrogen bond occupancy, we can see that JSK 199, which is active compound, has interact with 5 residue to stabilize the ligand, while JSK 106 has stabilized at 3 hydrogen bond. And we can say that SSN 585 actually overlap between them. For the MMPBSA calculation, which is for the binding free energy, we can look at the JSK 199 has a lower value, which indicate a good binding affinity between the JSK 199 and P84. And then compared to JSK 106, which is negative 31.92. For the visualization of P84 with JSK 199, we can see that JSK 199 stabilized at the pocket throughout the simulation, while JSK 106 is start to dislocate at 50 nanosecond and let the catalytic site, that color here, open. Due to that, we suspecting that dislocate of JSK 199 from the pocket, although it has a good binding affinity, but in the MG simulation, it clearly showed that JSK 199 was dislocated from the catalytic pocket. After that, due to we saw this dislocation, we want to investigate the binding affinity of BAA substrate towards the P84 in the presence of JSK 106. This structure was taken from the MD simulation at 35 nanosecond, and the binding affinity showed that it recorded negative 7.2 kilocalorie per mole. And the binding interaction as in the figure 10, we can see that histidine 471, aspartate 473, and aspartate 350, and cysteine 645, actually the catalytic site for the substrate, and it interact with the BAA. Okay, the possible event here is due to the P84 with GSK 106 stimulation was drifted away from the P84 pocket. This allowed the BAA substrate to occupy at the catalytic site. This might happen the huge difference in IC50 value between JSK 199 is active and JSK 106 is inactive. Another MD simulation is required to fully investigate the effect of P84 with JSK 106 with BAA complex. So in conclusion here, RMSD show that JSK 199 and JSK 106 is stable throughout the 100 nanosecond, and RMSF suggests that N3-month subdomain 1 each fluctuate more in the JSK 199 compared to JSK 106, and similar trend for the C-terminal was being observed. Higher hydrogen bond occupancy detected at JSK 199 compared to JSK 106 indicate a strong interaction between the P84 and active compound JSK 199. And the binding free energy JSK 199 suggests more favorable binding of the ligand P84 complex compared to JSK 106. Visualization show that JSK 199 is maintained at the P84 pocket, while JSK 106 is dislocated from the pocket. Binding JSK 106 did not have effect on substrate activity. It hoped that this work can help other researchers to discover more about the P84 non-complex inhibitor and answer the uncertainty regarding the P84 complex. Here is the enlargement for FRGS for this research grant, and thank you for me.